So here we are going to talk about that how the membrane proteins are synthesized in the secretory pathway on the ER membranes. So the first one is what we called as a membrane protein type 1. And here what you are seeing, that you are looking at the cytosol, that the cytosolic phase is the yellow, and the ER luminal phase of the phospholipid bilayer is green. You are seeing the transmembrane domain as the red-orange color, which is spanning the entire membrane of the ER, whereas you are seeing the N-terminal is in the ER lumen, and there is a cleaved signal peptide that has been shown, and the C-terminal is in the cytosol. So how this protein is basically synthesized. So the first thing that happens in this process, the ribosome right here is in the cytosol. This is your cytosolic ribosome, and it does have the corresponding mRNA that is going to be translated into a membrane protein type 1. So the first thing that happens, this is the channel through which the protein extrudes out of the ribosome as it is reading the mRNA. And the first thing that comes out right here, this is the N-terminal of the protein. And this is the signal sequence, or this is the signal peptide. This is a few amino acid with a certain sequence, and this is your signal peptide. This is the first thing that comes out. So as soon as this protein comes out, and this will be followed by, of course, the rest of the protein, that will be followed by right here, that the protein, and of course this will be the C-terminal of the protein, which will come out less. But what happens, fast forward, that the first thing, as soon as this signal peptide that comes out of the ribosome, when it was right here, immediately it will be basically covered by a protein which we known as the SRP. This is signal recognition particle. These are extremely large complexes made up of RNA and protein, and they are bound to GTP. They need the GTP to function, and they hydrolyze the GTP in this process and recharge by the GTP again. So we can just draw that GDP to GTP. So now, as soon as the signal peptide actually is covered by the SRP, that will cause something. That will basically, because the protein is, just think about that, that protein is very large RNA protein complex, is covering this part. It doesn't allow the ribosome to translate the protein anymore. And that we called as a translation arrest. Translation arrest. So this basically happened when the signal anchor sequence is covered by the SRP. Now what happens, this entire complex now comes to the ER membrane. So now if we just draw the ER membrane like this right here, these are the phospholipid on the cytosolic side. This is your cytosol. This is ER lumen. And you have this structure. Now, on the membrane of the ER, you have something which is called the SRP receptor. You have the SRP receptors. And the SRP will come over along with the translating mRNA, the protein, and the ribosome. This entire complex comes over and binds onto the SRP receptor. And here what you're going to see, that I'm just not drawing of the entire part, the GTP is going to be hydrolyzed to the GDP. And when that happens, the SRP dissociates from this complex, but before it does so, it basically hands over this complex to a structure which we called as a translocon. Translocon. This translocon is necessary, and they are present in the ER membrane to basically transport these protein through the ER membrane and insert it into the, so everything, transport the protein and inserting into the membrane is done by this translocon, very large complexes. So what's going to happen that the ribosome is handed over to this. So ribosome is going to sit on the translocon, and right here you have the mRNA, and how it looks like right here. This is your signal peptide. Now, interestingly, as soon as the SRP receptor releases the ribosome and the mRNA right here, translation will resume. So right here, translation resumes. So it will start translation back again. So first, the signal peptide, that is going to be inserted through here, and it will go into the lumen. This is followed by the sequence right here, and this is the N-terminal sequence, and we're going to see that again, and that will be followed by what we called as an STA signal. So STA signal is again stop transfer anchor signal. So as soon as the STA sequence appears, it will again stop translation. Stop translation. That means that basically the translation, basically think about that arrest of the translation is a correct word. Stop rather than stop translation. So translation is not happening in here. As this is happening, and why it needs to stop the translation, because you have to give time, the translocon, to allow this hydrophobic part, which is the transmembrane part, to diffuse out of the translocon into the membrane. And that is the time that is given by these interaction of the stop transfer anchor sequence. And now the entire part is going to be moving out from the translocon into the membrane. And you have the sequence right here. And as soon as it moves out, the stop transfer anchor signal sequence into the membrane, the rest of the C-terminal is going to be synthesized because translation will again resume. So that is how the membrane protein type 1 is synthesized. But it's not done yet. What's going to happen that right here, which is the signal peptide, that is now going to be cleaved by a membrane-bound enzyme, which is signal peptidase. So the signal peptidase is going to come over. This was the original N-terminal of the protein. It's going to cut the signal sequence, and now this will be the original N-terminal of the protein. And the signal peptide is degraded, but this will be the matured type 1 protein with the N-terminal in the ER lumen and the C-terminal in the cytosol. And that's how protein type 1 is synthesized. So in this video, we are going to talk about the membrane protein type 2. So the membrane protein type 2, as you can see, is in opposite orientation of type 1. So what that means is that what you're going to see, the C-terminal is going to be in the endoplasmic reticular lumen, and the N-terminal of the protein is going to be in the cytosol. So this orientation of this is exactly the opposite of the type 1 membrane protein. And we're going to see that how this is actually synthesized. So there is basically a little bit of difference in between that. So let's draw the membrane, that how it is in here. So let's draw that is the upper leaflet, and this is the upper leaflet of the ER membrane. Right here is the orientation, and this is your cytosol, and this is your ER lumen. So now what is happening right here is the same way. You have the two ribosomes, ribosomal subunit, you have the mRNA, and you have the protein is trying to get synthesized. Now, interestingly, that how this is basically going to happen, the first, you have this protein, the N-terminal, this is coming out. Remember that there is no signal sequence in the N-terminal. But what happens that after some time, there is an area where there is a few amino acids which are going to come out as well. These are positively charged amino acids. That will be followed by a sequence which is hydrophobic. So just think about that. That hydrophobic sequence is going to come out. As soon as that hydrophobic sequence comes out, so we can just modify this picture just a little bit. So as soon as that comes out, what's going to happen? We're going to modify this picture a little bit to understand the next step. So here, this sequence is now out. This is the N-terminal sequence, and we have some plus charges amino acid right here, plus charge amino acid. Now, this sequence right now is known as the SA, or the signal anchor sequence. This is not STA, which is a stop transfer anchor signal. So signal anchor sequence is basically the signal for the SRP to bind and bring that ribosome mRNA complex onto the ER membrane as well. So what's going to happen? The same way, this is going to be recognized by the SRP. And as you can see, the SRP and the GTP, the same way is involved. It's going to bring this entire portion onto the ER membrane. So I'm just not drawing the other parts. So I'm just drawing the ribosome that is sitting on the ER membrane right here, small, large subunits, and then you have the mRNA. And what we are going to see in this process, very interestingly, let's draw the translocon as well. So I'm just drawing the translocon as this structure right here. This is your translocon. So what's going to happen that, as you can see, this N-terminal, there is no signal sequence, but there is a plus charge right here. And what happens that as this protein is synthesized, these plus charges are going to be repelled by some of the plus charges that is already present on this surface or the face of the translocon. So what's going to happen that the N-terminal is right here and the plus charges are just right here. That is not going to go into the lumen of the translocon. So now what is happening? Signal anchor sequence that is basically dropped in here. It is basically, it's working its way with the translocon. After this, what's going to happen that you can see that right here, I'm just drawing in a different part. This is the C-terminal portion of the protein. And this entire U-shaped portion, whereas this is interacting kind of bound with the translocon, whereas this portion is synthesized and this U-shaped structure, think about that, this U-shaped string is being basically being dropped into through the translocon into the lumen. So ultimately this entire portion is going to be dropped over. So what you are going to see after that this hydrophobic portion then is going to transferred into the membrane. So what you will have you have the N-terminal right here. So this is your N-terminal with these plus sequences, and your C-terminal, which is gradually moving in as it is lowering this U-shaped. So the C-terminal will be dropped through the translocon, and the hydrophobic portion, which is in the middle right here, this is the transmembrane domain. That's going to eventually diffuse out from the translocon into the membrane, and that's how your type 2 protein is synthesized. N-terminal in the cytosol, C-terminal in the ER lumen. So that's how the type 2 membrane protein is synthesized. Now let's talk about how the membrane protein type 3 is actually synthesized. And This protein is synthesized in a very similar manner how the type 1 is synthesized, but there is a huge difference in between the type 1 and the type 3. The orientation is the same, but there is no signal peptide. As you can see that right here, you have the N-terminal in the ER lumen, then you have the transmembrane domain, and then it's followed by the C-terminal, and there is no signal peptide. So how this is essentially being synthesized. So what happens, let's start with the same process. You have the ribosome right here, and it's reading its mRNA, and you have the first portion of the N-terminal that extruded out. I'm just rather than just drawing two pictures, just one is good enough. So N-terminal is coming out. This is happening in the cytosol, N-terminal, or before that, there is no signal. But after that, what happens? After that N-terminal, this transmembrane hydrophobic signal appears. And as soon as this appears, again, as you remember, that we will call this one as an SA sequence, which is again signal anchor sequence. So the signal anchor sequence will appear, and as soon as this signal anchor sequence appears, again the SRP is going to come over and bind onto that, and that will bind in here. This will be followed by a sequence which is plus charged right here. So what's going to happen that when this entire portion is actually coming onto the ER membrane, you have the ribosome sitting right here, and you have the translocon right here. That's your translocon. And now, if you carefully look at that, this N-terminal doesn't have any plus charges on those. So what's going to happen that the N-terminal, it can go through right here, it can go through the lumen of the translocon, basically, the transport channel. And then the hydrophobic sequence is going to bind right here with the interaction, hydrophobic interaction with the translocon proteins. And then the C-terminal is going to get synthesized, and these are plus charged right here. Plus, there are some plus charge residues that are going to come over in the C-terminal. And due to this C-terminal is not going to enter into the lumen of the translocon either. So this is going to remain on the outside or on the cytosolic side. So that is the C-terminal. So eventually what's going to happen that after this brief interaction, the entire protein is going to move into the membrane. Here you have the N-terminal, and this term you have the C-terminal. So very straightforward. The only thing is that right here, the signal anchor sequence is bringing the ribosome and the translating ribosome from the cytosol onto the membrane. And this is also done by the SRP at the expense of GTP. So that's all about protein type 3. So the next what we are going to see is that how the tail-anchored proteins are basically synthesized. And this is synthesized in a little bit different way, actually very different way compared to the other membrane proteins or the other secretory membrane proteins. So here you can see the C-terminal is buried into the transmembrane domain, and the N-terminal is in the cytosol. And let's take a look at that, how this protein is synthesized. So the main problem for this synthesis is the sequence or the signal sequence. So right here you have the mRNA, and the ribosome is reading the mRNA. But the problem, what happens, that this is the N-terminal, and it is being synthesized by the ribosome coming out, but the signal is all the way in the C-terminal. This is the C-terminal, which is basically the transmembrane domain as well. And this C-terminal will not appear out of the ribosome until the entire protein is synthesized and released into the cytosol. So for tail-anchored protein, the entire protein is synthesized in the cytosol. So basically synthesized in the cytosol. So this is one of the important things. But then how you bring this protein to the ER membrane? That's the question. Now, before that, also we need to address one of the important part. For all secretory membrane proteins that are synthesized with the ribosomes on the ER membrane, all of them are basically doing what is called that co-translational translocation. That means that all the other type, membrane type 1, 2, 3, that what we have seen, and we're going to see the other parts as well. As the protein is translated, the protein is translocated through the lumen of the translocon, and the translation, this is done by the ribosome, and through the lumen of the translocon in this process. And it is co-translational. It is happening at the same time. The ribosome sitting on the translocon and doing the translation, simultaneously the translocation is happening. Of course, it will stop at the stop transfer anchor sequence and all those, but it is that. But here you can see the entire protein is synthesized in the cytosol. Now, after it is completely synthesized, now it will be brought onto the ER membrane. So this whole thing changes only for this type of membrane protein, which will be the post-translational translocation. And this will be the main difference between that. So just make sure that you just remember that particularly for the tail-anchored protein, the post-translational translocation occurs. What are the other differences? There are some other differences as well. So the next one is that when the C-terminal comes out, and you know that this is a transmembrane, so this is hydrophobic, and this is not stable in the aqueous environment. So immediately a couple of proteins actually basically get involved in this process to cover that, and we call this as a sequestering, just hiding that part. And these three proteins are SGT2, GET4, and GET5. So these three proteins is GT2, GET4, GET5. These three protein complexes cover that hydrophobic part so that it doesn't interact with the aqueous environment and stabilize the protein in the cytosol, because it is made in the cytosol. Now the transport protein comes in, which will take this protein to the ER membrane, and it is not SRP, and it is a different molecule. This molecule is known as the GET3 complex. So this is your GET3 complex, and it works with the ATP. So when it is charged with the ATP, it is functional, and now this GET3 complex will replace these complexes, which are the SGT2, GET4, and GET5, and the protein will be picked up by GET3. GET3 is going to sequester this protein so that it is still not exposed to that, and you have the N-terminal here, you have the C-terminal here, the protein is protected by the GET3. And the rest is similar, but it is not bringing the ribosome in the picture as such. So right here you have the ER membrane, and here you have a particular receptor, which is the GET receptor. You have the GET receptor. And the same way that the SRP receptor gets it, the GET3 and the protein, this is going to be transferred here. And once the GET3 complex get into this process, what it is going to do, that this will trigger the hydrolysis of the GTP to GDP. remember that this is happening when it is sitting on the get receptor, basically. So it already brought the protein. At that time, when it brought the protein, here you have the translocon right here. And once this dissociation happened, this protein is basically inserted into the translocon. And right here, this is the N terminal, and this is basically the C terminal. So here what happens, we can say that this is basically the signal, which is the SA or the signal anchor sequence. Remember that signal anchor sequence is the signal which brings a protein to the ER membrane, but here the signal anchor sequence is at the C terminal, and that's why you need to do a separate process which is basically synthesizing the entire protein in the cytosol and bring it back. Because unless the C terminal is appearing out from the ribosome, there is no way that it can be recognized. The signal anchor cannot be recognized by the proteins, so that's why it is completely synthesized in the cytosol. And eventually this will diffuse out into the membrane, whereas this is your N terminal and this is your tail anchored C terminal inserted into the membrane. And this is how the tail anchored proteins are synthesized. Next, we are going to talk about the GPI anchored proteins. So the GPI anchored proteins are membrane proteins which are anchored into the membrane with the glycophosphatidylenosydyl inositol anchor, which is a membrane lipid. And let's take a look at that, how they are going to be synthesized. You can see that right here the C terminal is blunt. It is inserted into the membrane. N terminal is in the lumen. And how this whole process actually going on, we are going to see that. So this essentially is very similar to how proteins are synthesized in the type 1. So which is not shown here is right here. You have the ribosome and you have the mRNA, and there is no problem with this. Right here you have the signal peptide. You have the signal peptide exactly like type 1, and it is followed by the protein, which is basically like this, the protein part, and this is your C terminal. And the SRP, of course, and the GTP is involved in this process, and the signal peptide will be cleaved, and this protein will be in the same orientation as the type 1 protein. So if we just look at the orientation of the protein into the membrane, let's take a look at that, how this basically happens. Right here is your cytosolic leaflet. Right here is your ER leaflet. This is your cytosol. This is your ER lumen. So the protein is inserted into the membrane like this. So let's take a look at this, and then you have the hydrophobic sequence. So right here, this is the hydrophobic sequence of the protein, and I'm just not drawing the translocon. I can just, of course, do right here. So the translocon mediated as well, same process. So right here is your C terminal of the protein, and right here is your N terminal right here. And what is not been shown is that there is a signal peptide that is cleaved. So you have this situation of the protein, N terminal, and right here is the C terminal, and there is no C terminal, and there is no cytosolic extension of the C terminal. So now what happens, that this is very interesting, that this entire protein is not used to be added onto the glycophosphatidylenosydyl inositol anchors. So what happens that this glycophosphatidylenosydyl inositol anchors, these are present as the membrane lipids. These are already preformed, and there is an enzyme which is trans-GPI. It is known as the GPI transamidase. So what happens that it will cleave. It is the protein is actually cleaved, which is the luminal part, and transfer to this glycophosphatidylenosydyl inositol anchor, and right here it is covalently linked. So this is the N terminal of the protein, and this becomes the new C terminal of the protein because it is cleaved from here. And right here, this is your GPI anchor. So now you can see that the N terminal is this way. Right here, your GPI anchor is in this way. Now the question is that what is the problem of this protein being in the plasma membrane versus this? The reason for this is actually versatility. So what happens when the hydrophobic part of a protein is, as an integral membrane protein, is inserted into the membrane, it does have some restriction in terms of mobility, and it can just go all over the places wherever it wants. But on the other hand, the GPI, particularly glycophosphatidyl'inositol anchors, they have high affinity for some specific region, high affinity, say for an example, lipid rafts. So GPI anchored protein have a high affinity for lipid rafts. So what's going to happen that if a protein is associated with a GPI anchor, it will find out the lipid rafts and these microdomains, and these proteins will be placed in that microdomains. It is much more easier for a lipid molecule to move in the lipid bilayer to find the lipid rafts compared to that if it was a transmembrane protein and to find the microdomains or the lipid rafts. That's why it is pre-synthesized this way, then it is cleaved off and transferred by the transamidase into the GPI anchor. So the next what we are going to see is the membrane protein type 4, and the type 4 is a multi-pass protein, and it is also subdivided into 4A and the 4B. In a 4A, we see the N and the C terminal are basically on the same side. Here you can see that the N terminal and the C terminal, they are in the cytosol. They can be in the ER lumen as well. So basically it will be the same in the reverse order. And these are synthesized in a little bit different manner, and also, of course, SRP-GTP is also involved. So the first thing is that when you look at the 4A, usually they will have an even number of transmembrane domains, and that allows the N terminal and the C terminal to end up in the same side, either the cytosol or the ER lumen. So let's take a look at that first, how this is synthesized. As you can see, that right here is your ribosome, and you have the mRNA. And first, what's going to happen, and I have to draw a couple of ribosomes to do this. So the first, the N terminal actually comes out. There is no signal in the N terminal. Right here, the N terminal comes out, and there is no signal in the N terminal. And after that, what happens, that you have a hydrophobic signal that comes out. But before that, what you have is some of the plus charge amino acids right here. So now what happens, that you have this first sequence of hydrophobic sequences comes out, and this is your SA. This is your signal anchor sequence. So as soon as you see the signal anchor sequence, that means that it is the signal by which the SRP will come over and bind here and bring this onto the translocation to be inserted into the membrane. So how this basically happens now. So let's take a look at the membrane right here. You have the two leaflets of the membrane. Now you have the translocation right here. Translocation, and the membrane is right here. It is inserted. Now remember that we already talked about this N terminal being in the cytosolic part. Because due to these plus charges, the N terminal is not going to be lowered into the lumen, so it will stick out into the cytosolic side. Whereas right here, this is going to synthesized. Right here, this is your SA sequence. After the SA sequence is synthesized, so this is where the ribosome is sitting on top of the translocation. So this will be followed by a synthesis of another loop-like structure, And this is basically the luminal part or the loop in the ER lumen. So that's going to be synthesized after this. After that is a sequence. And after that, there is another thing is going to happen. After that, another hydrophobic sequence is to come out, and this is basically the protein. And that hydrophobic sequence is going to be on the other side right here. But this hydrophobic sequence is not the SA sequence. That's going to arrest the translation, and we call that a stop transfer anchor sequence. Stop transfer anchor sequence. And why this is stop transferring? The reason is that you have to give the translocon a little bit of time, so that these two hydrophobic sequences, transmembrane domain, can move out into the membrane. And that's the reason that the stop transfer anchor sequence, as soon as it appears, it interacts with the translocon, it essentially doesn't allow the ribosome to push the polypeptide furthermore. So it's going to be now transferred. So how this protein will look like, the same protein, once it is inserted into the membrane, it will look like this. And you have right here the two sequence. One is SA, another is STA. And right here, these are your plus, and this is your N. So the STA is now in the membrane. Now what's going to happen, that as the STA and the SA sequence moves out, ribosome move out of translocon. This is very different from what happened earlier. The ribosome moves out. And if the ribosome also moves out, the rest of the protein is going to be synthesized by the ribosome, which is hanging in the cytosol. It is not on the translocon. Now, it will use the translocon to move the protein. It will use the SRP and GTP to move that, but the ribosome is going to hang around in the cytosol. The reason for that why we needed the ribosome to sit on the translocon in the very first place, because we needed to put this protein in here, but now the protein is anchored or tethered into the membrane. This membrane is not going to go anywhere. So the rest of the protein, if it is synthesized by the cytosolic ribosome, and if SRP brings the next hydrophobic portion into the translocon, there is no problem with that. So now what you are going to see, that you are going to see this synthesize this, and again there will be some plus sequences, and these plus sequences means that this will not go into the lumen of the translocon. This will be followed by another signal anchor sequence. And why we need another signal anchor sequence? Remember that the ribosome is floating in the cytosol. When this hydrophobic sequence comes in, this protein needs to go somewhere. So the SRP binds to that and brings this entire part, puts this signal anchor sequence into the translocon, but doesn't put that plus into the translocon. So it remains in the cytosol. So this is the signal that needs to be brought. And that will be followed by the loop right here. And that will be followed by the STA, stop transfer anchor. And why again? Because remember that you have to give the translocon the time to move these two diffuse into the membrane. And that is the time that is basically given. So, and this will be the C terminal. So this is basically how it is being synthesized. And what you are going to see, that if you have even number in 4A transmembrane domains, your first signal is going to be signal anchor, followed by an STA, followed by an SA, STA. If you have 10, 12, even 16 transmembrane domains, your SA and STA sequences are going to basically alternate. That's basically what happens. And at one point of time, the SA and STA both are going to be inserted into the translocon. This both are going to be inside the translocon, and so on. And again, remember that signal anchor sequence is bound to the SRP, which brings that hydrophobic part to be inserted into the translocon, whereas the STA, the stop transfer anchor sequence, interact with the translocon and arrest translation so that it gets the time to push these two hydrophobic sequence into the membrane. And then once it's in the membrane, then the rest can actually presume. So that is your type 4A. So the next, what we are going to talk about is the type 4B membrane protein. This will be a multi-pass protein as well. And here, the N-terminal and the C-terminal is going to be on the opposite end. So here you can see that the N-terminal is in the ER lumen and the C-terminal is in the cytosol. And it could be in the reverse order as well. N-terminal can be in the cytosol and the C-terminal can be in the ER lumen as well. And here also SRP-GTP is of course involved. The interesting part is that unlike the type 4A, where you have an even number of the transmembrane domain, in order to put the N-terminal and the C-terminal in opposite end on the opposite sides, which means like one cytosolic, one ER lumen, you will have odd numbers of transmembrane domains. So here you can see that right here, 1, 2, 3, 4, and 5. So it can be 3, it can be 5, 7, and so on. But it has to be an odd number to put the N and C terminal on the opposite ends. So how this again happens? This will be a very similar way that how the type 4A synthesize, just a little bit difference right here. So here you can see that the ribosome right here, and you have the mRNA, and the N-terminal doesn't have any signal. So what will happen? This part of the N-terminal is going to be synthesized in the cytosol, which will be followed by right here, the signal right here. So what's going to happen that right here, this is the signal, which is the SA sequence. And this will be followed by a portion of plus-ended plus charge amino acids will follow that. Now what will happen, interestingly, this SA sequence, this is where it is a little bit different than the type 4B. So here, what we are looking at that your translocon is right here, and you have your N-terminal in here. So after that, this will be followed by the SA sequence, signal anchor sequence, that will interact with that. Now, that will be followed by a loop, which is essentially going to be a plus charge. That means that this portion is not going to go into the lumen. And at this point, what happens after this plus charge is coming out from here, the ribosome is actually dissociated. So it doesn't sit there until there is a stop transfer anchor sequence comes out. Ribosome dissociates right here. So ribosome doesn't sit in there anymore. And then you will have the next, this is your first, this is your second, so I just write 1. You have another plus sequence coming out, and the ribosome is in the cytosol. And this is happening as the ribosome is synthesizing that in the cytosol and right here. Now, after this, followed by that, you will have another signal that will come out. So you will have another signal, hydrophobic signal, that will come out. That hydrophobic signal is again going to be another signal anchor sequence. The reason for that is you are going to use another translocon right here and going to put this in here, which is the signal anchor sequence. And why this is so, and why this is not the same case in the type 4A. So in the type 4A, what happened, that as you could see, the N-terminal was on the cytosolic side, and it synthesized the signal anchor sequence, and after a loop, the stop transfer anchor sequence goes up. Here what is happening, you have the N terminal in the lumen, the signal anchor sequence is going up, and then you have these plus charges right here. So the next one is going to wander around, this hydrophobic sequence is going to wander around in the cytosol, if not brought in. So once this comes in, you still again need an SRP or SRP-like proteins to bring that sequence or the hydrophobic sequence into the lumen of the translocon. And that is the reason that as both of the sequence, the first one and the second hydrophobic sequences, both are required to bring that part onto the translocon, both are signals, and we call them as a signal anchor sequence. And after that, they will alternate. So what will happen now, you have the loop, and after that, from the loop, so let me draw it this way, it will be the loop, and now the next hydrophobic sequence is inserted the same way right here, and this will be an STA, stop transfer anchor sequence. So after this is formed, they will wait a little bit, the ribosome will stop translating so that this entire portion, the first one already did, and this portion is moved into the plasma membrane, and then again this part is going to be synthesized, which are not going to get into the translocon, followed by another SS sequence, followed by another SS sequence, followed by another STS sequence. So the only difference is that the first two sequence, 1 and 2, they are going to be the signal anchor sequences in type 4B, and then the signal anchor STA, SA, STA is going to go all the way in. And the membrane topology, how basically you will know that how is the membrane topology. If we just draw the protein as a linear protein, you can see that this is the N terminal, this is the C terminal. What you are going to see that you have a hydrophobic signal right here, hydrophobic signal right here, hydrophobic signal, and all you have to see is basically right here, you have the plus charges right here, plus charges right here. So the reason is that wherever you see the plus charges, it will be the cytosolic side. This is ER lumen, this is the cytosolic side. And then this is followed by the ER lumen, followed by that. So whenever you see a side of a SA or a signal anchor sequence does have a plus sign, you know that that portion of the loop or the protein is going to be on the cytosolic side, because that is not going through the translocon. So this is how the type 4B is synthesized.